Hello, welcome to my Genetics 311 Module 4 exam video. Today is going to be talking about characteristics of DNA. So a nucleotide is the base unit of DNA and it has three parts. So we're going to go in detail to these three parts. So the first part is the nitrogenous bases. And these bases have two categories, pyrimidines and purines. Pyrimidines are cytosine and thymine, and these have one ring. Purines have two rings, and that is adenine and guanine. So these are complementary to each other. Guanine is complementary to cytosine, and they form three hydrogen bonds in between them. Adenine and thymine form two hydrogen bonds between them, and they pair up together to make that double helix of our DNA. So next is going to be the phosphate backbone. So this is the hydrophilic interaction that's going to keep water out of our middle section of DNA. So what happens is we cleave off two of these phosphates, and they're going to bond to the 3' carbon in order for us to get this very tight phosphodiester bond. So next is going to be a ribose sugar. This has 5 carbons on it. The first carbon is going to be what attaches to our nucleic acid base. So this is going to help form that full nucleotide. The second one is going to determine DNA from RNA. If it has an H, it's going to be DNA deoxyribose. And if it has an OH, it's going to be a ribose. So that's RNA. Our 3' attaches to phosphate groups below. So it's going to help form that phosphodiester bond chain. That's going to keep the water out from our middle section. And then our 5' carbon is going to do the same, but it's going to be up top and it's going to attach to the next phosphate group above it. So DNA strands have base stacking. So these are organized in very specific ways so that they minimize the forces that are trying to pull our DNA apart. It's a very big stabilizing factor. And our strands are anti-parallel. So one way is going to go 5' to 3', and the other way is going to go 3' to 5'. And this is going to maximize our base pairing and make sure that our DNA stays together. So grooves, we have major and minor grooves. Minor grooves are going to be a lot smaller than our major grooves, and these are going to be important because they help proteins identify different regions of our DNA for molecular processes. So handedness, DNA will either be left or right-handed. If it's going up, our helix is going up and to the right, it's going to be a right-handed DNA strand, and if it's going up and to the left, it's going to be a left-handed DNA. So over here we have an example. This is going to go up and over to the right, so this will be a right-handed DNA sequence. So DNA, when it's put in a salt solution, how is it going to affect the melting point and how fast it'll denature? So with no salt in a solution, there's a lot of negative charge in our phosphate backbone. So that's going to be pulling apart our strands, and it'll be easier for it to denature, so a lower temperature of melting. When we put it in a high salt solution, these ions are going to neutralize these negative charges. And that's going to cause less forces to be pulling our DNA apart, and it's going to be more stabilized. So a higher salt concentration is going to lead to a higher temperature of melting, which is going to make our DNA more stable. So here I have some of my references, and thank you guys for watching. I hope you enjoyed.